Thank you. Mike Christoffers and Weed Genetics here at NDSU, and I'm coming to you from my lab today because I'm going to show you a couple things, something that I couldn't do in previous Wild World of Weeds workshops. So you can start with the next slide, please. And so I'm just going to update you on some of our research. Here is a slide showing kind of the end goal. So some of the work we're doing is for this new, very emerging, not yet released type of pest management. It's not just for weeds, called the gene drive, which I have talked about a little bit before, but I do want to briefly go through how this works. One type of drive that we are working with is called a sensitization drive, and it would be, perhaps if it works, a way to reverse herbicide resistance back to susceptibility. Basically, a gene drive is a way to spread a gene that we want through a biological system, and it's being looked at among researchers in all types of pest and pest control. So very basically how a gene drive works is it biases the inheritance toward something that we want. If you think of herbicide resistance, there we're selecting for something we don't want, the resistant types. Here, just like if we were flipping a coin, you know, we'd expect 50% heads, 50% tails. With the gene drive system, we're trying to bias that as if you were having a double-sided coin, which is going to greatly enhance your chances of getting the heads, right? And kind of the driving pest in this is mosquitoes. And in particular for human health and the fact that they are a vector for malaria. So groups such as Target Malaria, an international group, is looking at gene drives as a way to either make mosquito populations, especially the mosquito species that carry malaria, make them either unable to carry malaria or to try to reduce the populations of the mosquitoes themselves. Another area of pest control being looked at is for rodents. So an example there would be in New Zealand. They have an initiative for what they call predator-free 2050, try to reduce the number of mammalian predators that they have in New Zealand. Some of the more recent research shows that getting a drive or this type of biased inheritance to work in mammals is more difficult than it's been with insects such as mosquitoes. But there have been some recent successes in spreading really infertility through mammalian populations. Not of course released into the wild, but in the laboratory at this point. So spotted wing Drosophila, or fruit flies, a important agricultural pest for fruit production. And that's also one of the targets researchers, especially out in California, have been looking at for gene drives. And they're working with a system called Medea, where high temperatures, so the flies exposed to a high temperature in the environment would be lethal to them, but they would still reproduce at a lower temperature in order to spread the drive. So it's kind of a balance. You don't want a drive to just kill the pest species. You want some reproduction so that your gene of interest gets spread. And then in 2016, there was a National Academy of Sciences report, which went through several scenarios of how gene drives could be applied to pest control, and Palmer amaranth was on that list. And the example they gave was a gene to bias the sex ratios, for example, to make more of the plants male instead of female to decrease seed production. Pat Trennell, University of Illinois, is working on the genetic sequences that cause male versus female in both Palmer amaranth and water hemp, which might be a target he suggests for gene drive research and perhaps something into the field in the future. Now we are working on water hemp. We are working on the very basics of getting information that we would need in order to see how successful a sensitizing drive would be to revert herbicide resistance to susceptibility. And so basically this, I'm just repeating the same slide showing what we are working towards as an eventual goal, to spread susceptibility back into water hemp populations. Now, I'll talk more specifically about the gene target in a minute here. So how do we do this? For this type of research, it is important to work on tissue that is not going to escape our experiments or escape laboratory containment. So the first thing we did was to develop a tissue culture system. And so the way that works is that we germinate the water hemp seeds, as you see on the left. We remove a part of that stem, which is called the hypocotyl, and then we grow it on a culture media with hormones and food for the cultures, and we get this growth of cells. And so let me show, if it shows up here, this is then one of our bottles with, you see the water hemp cells growing on the inside there. It's just undifferentiated mass of cells. But we know that this type of tissue culture in the literature in the past has been shown to be susceptible to herbicides. And so the data I'm going to show you today is about how the cells that we're working with, they are susceptible to group 2 herbicides or ALS inhibitors. So what do we do with that mass of cells? We want to be able to have tissue that we can easily get the herbicides to, so it's better to have individual cells rather than a mass of cells that only exposed to media on one side. And so what we do is we take that mass and we put it into liquid. And here is a culture of water hemp. These are water hemp cells, but they are more individualized. There are still some clumps there. And, you know, we have the same hormones and food in here, and we can keep these growing indefinitely. We just take a sample and, you know, put the cells into new media every once in a while. So those types of cell suspension cultures is what we are working on right now for the herbicide work. And so this shows you a microscopic view of what those cells look like. So when we first take that mass of cells and put it into the cell suspension culture, we have still some clumps, as you see in the first microscopic view there. And then as we culture and subculture these cells, we do get more individualized cells, which you see on the views below. And so this shows you an increase in the growth over just short of 3 weeks. And so the way we measure the growth is just how dense these cultures are. When we first put the cells in there, we have a lot of clear liquid, and then as the suspension grows, it gets cloudier. And so this OD 600 or optical density is measuring that cloudiness. And so over the course of about 3 weeks or so, we can get the increase in growth. And so what we wanted to do was to see if we could get a decrease in this growth with exposure to herbicides to make sure that the cells are responding as we thought they would. And so this is thiophensulfuron methyl as an example. So this would be like Harmony herbicide. And we're learning more about how to grow the cells. So you notice our first run wasn't as good as the second as far as the growth. But we do see in the first run, as we increase the concentration, now this is a nanomolar, so a very small amount of the herbicide actually. As we go from 0 and then increase the dosage, we do get less growth as we would expect, and especially see that in the second run, the second time we did it. So at the 0.01, we still get growth very similar to not having the herbicide in there. And then as we increase the herbicide dose, we get less and less growth. So this tells us that the growth of the water hemp cells in culture is responding to herbicide. And so imazethapyr and other ALS inhibitors in IMI, group 2, such as Pursuit. And again, we didn't get as good of growth on the first run, but we're learning more and more about how to do this, as I said. And we again see that the growth of the water hemp does respond to the herbicide in reduced growth. All right, so back to the current status of gene drives. Well, for us, now we have a system that we can change the genes. So with gene drive systems, we need to change genes such as resistance to susceptibility. The cultures we're working with right now are susceptible, which is what you saw in the figures. But we need to develop a transformation system to get the components of the drive into the cells. And so that's what we're working on now. Why did we choose ALS? Because first of all, that gene for group 2 herbicides is very well studied. It is very well studied in CRISPR-based gene editing, which is the system now that has really allowed gene drives a chance to work. Gene drives were proposed earlier before we had CRISPR-based gene editing. But CRISPR is really the direction everyone's looking at right now. And then ALS is also well studied in terms of herbicide resistance. So we know the mutations there that we can change. As far as gene drives overall, a few things have been happening very recently. One of the concerns that we've had was the control of a gene drive system, how not to get it out of hand, I guess we could say, and also to perhaps eliminate the system when it's not needed anyway. And so a group of researchers have been working with such systems. There's one called eChaser, which will inactivate the gene drive system. And then there's another one they're calling Eraser that will actually, looks like it can take the gene drive or eliminate it from the pest organisms when it's not needed there anymore. And right now it's not perfect, but that's where the research is going. As far as field release, so again, those working with insects are kind of in the lead in this. And so they're working on really guidelines for eventual field testing, which has not been done yet. Everything has been in the lab or for insects for cage experiments. Another thing that's being looked at is public support. And so North Carolina State and University of Wisconsin did a survey recently about public support for gene drives for pests in agriculture, especially looking at insects. And the support is about 50-50. So if you take about 50% fairly supportive of gene drives and then add to that another 30% who aren't quite sure yet, that there is public support out there. And these efforts have been, it will probably be public rather than private type of releases into the environment for pest control. So with that, I will see if there's any questions or if there's time for that. The next slide does have some acknowledgements, and my technician has done a lot of this work, and then some organizations that are funding this research or research related to it.
Appreciate the talks. Pretty innovative and fun there to see you showing us the methods in real time. So something maybe we'll keep going into the future here for some meetings. Might have a question coming in here. Yep, we do have one coming in here. All right, will we be looking at other herbicide groups to develop susceptibility to? So you've showed ALS inhibitor herbicides, or we're looking at other herbicide groups?
Yeah, so again, why we're looking at ALS is because it's well studied, and to see how well the system works in an organism like water hemp. That's where we started. Something like glyphosate resistance, for example, that tends to be an increase in the number of gene copies. That would be more difficult type of resistance to revert back to susceptibility, because you've got all those different gene targets. Now there is research, not in weeds, but in other pests, looking at getting rid of many gene copies, but it is more difficult. So that's why we started with ALS. But really, it's applicable to anything that's genetic, which most cases of resistance that's inherited would be.
All right, thank you.